Hello everybody and welcome to VTT webinar. Today we're covering the production of sustainable next generation biocolours. My name is Michael Healy-Wren and I'll be your host. I'm Business Development Manager responsible for industrial biotechnology research here at VTT, and it's my great pleasure to welcome you to today's webinar. Just a little bit of housekeeping as we get started. During the webinar we'll have a Q&A. And the overall time of the webinar is around 50 to 60 minutes. For the Q&A, please submit your questions through the Ask a Question tab in the chat. Please note that the tab is not always visible in full screen mode, and we'll answer your questions at the end of the webinar, so we'll have a facilitated Q&A section later on. And we'll also provide you with the webinar slides and the recording about a week after the live webinar, so you'll get all the materials shown today by our great speakers. So the agenda today is we have a brief welcome from me, and then we'll be moving on to looking at sustainable anthraquinone pigment production with a SynBio approach by Jörg, one of our research team leaders here at VTT. Then Karen, who's the founder and CEO of Vienna Textile Lab, will take us through microbial pigments as an alternative to synthetic dyes in the textile industry. And then we'll finish up with Iro's presentation talking about fashion for goods work from lab to mill, evaluating and scaling biogenic colorants. Following that, we'll have a live Q&A, and then there'll be a kind of quick summary at the end of today's session. So I just wanted to say a couple of things very quickly before we get started. So as you know, this is actually the second one of these biocolour webinars that we've had in a series that VTT has been organising, where we've been inviting some of the main players in this industry to take part and share their research and the amazing work that they're doing with startups and research institutes. And the reason why we're so kind of interested in this is that we think the sustainable colorants really matter. Most colorants are petrochemical based, and it means that they have a really high environmental impact. And because of that, there's really increasing regulatory and consumer preferences for safe and bio-based solutions. Natural alternatives, though, are actually really struggle with scale and cost and performance. So we believe that biotechnology really enables a scalable, high-performance, sustainable solution for a range of different applications, so from food, cosmetics, to textiles. And we really believe that sustainability and performance must go hand in hand. So that's enough from me. I'll now pass on to Jörg to give his presentation and look forward to the Q&A. And please don't forget to put your questions in the box.
Yes, thanks for the great introduction to the topic, Michael. My name is Jörg de Ruiter. I'm leading our production host engineering team at VTT, where we base a lot of our biocolours work in. And the topic I'll be talking about today is anthraquinone pigments produced by Cortinarius type mushrooms. These mushrooms, shown here in the background of the picture of the slide, grow, for example, in the Finnish forest, but also in many other forests in Europe. They have been traditionally used for dyeing applications because of their intense yellow to red colors and excellent stability. Additionally, biosafety and environmental toxicity studies are available for them, making it easier to take them into use into real processes. However, when we tried to grow these mushrooms in the lab, they didn't produce any colors. It was remarkable because this approach is something that has worked for us with a few different mushrooms that produce interesting pigment molecules. However, in this case, whatever we tried, the mycelia that we grew in the lab just stayed white. So therefore, we opted to go for a synthetic biology approach. For this, we made genome and transcriptome sequencing data from conditions where color was present, for example, mushrooms grown in the forest on the top, or absent, our mushrooms grown in the lab. And then we compared these different profiles to select the genes and other things that we expected to be part of the anthraquinone production pathway. And then based on this selection, we started to make our yeast expression strains. We're using Baker's yeast, Saccharomyces cerevisiae, for this because it's a very easy to work organism within the lab. So with this slide, I just quickly wanted to show that once we have targeted a polysaccharidase synthase or PKS that we expect to make this anthraquinone scaffold, and we start actually looking in our genome data, we can find quite a lot of relevant other putative genes in the gene clusters. So for example, as you can see here, we find monooxygenases, O-methyltransferases, and oxidases, which are all potential enzymes that could modify this anthraquinone scaffold further. So what we knew from the literature and our own first bit of research is that a polyketide synthase converts the primary metabolites into the first two stable intermediates through a few steps, and these intermediates are endocrossin and amodin. They are both yellow, and they only differ in the carboxylic acid group in the top right of the molecule in this picture. Then we need action of one O-methyltransferase to create either dermolytin in the middle top or fischion in the middle bottom. These stable intermediates are also both still yellow. And then finally, we believe that through action of an oxygenase, either one or two reactions, we get the final red stable compounds, which are dermorubin and dermocybin. So we started from the beginning of the pathway and expressed our putative PKS enzymes in our yeast. And right away, we could see color formation happening in our colonies, both in liquid and in solid media. So, of course, you think we're making a yellow compound, we're expecting the yeast to turn yellow. Well, yes, in an ideal world, it would be the case, but we could at least see here a clear color change from, for example, on the right picture, the negative controls are quite white and the anthraquinone producing strain is quite brown, and the same for the liquid cultures on the left. So to make sure that we were producing really the right compounds, we then developed an LC-MS method to functionally validate this PKS enzymes. So with this information, we were now able to show that we have strains either expressing PKS2 from our forest mushroom or PKS3 from a genome sequence of a related species were producing both amodin and endocrossin. So we had the start of our pathway and discovered two novel enzymes there. Then for the next step, we were looking for our O-methyltransferases. So we tested 6 O-methyltransferases, and from this we found 2 that produced dermolytin from endocrossin, here being marked with green on the LC-MS spectra, being OMT2 and OMT5, and 1 enzyme that produced fischion from amodin, being OMT4, here shown in pink on the LC-MS data. So now we were able to produce the second stable yellow intermediate in our yeast strain, and that really nicely shows the value of our SynBio approach. Because thinking about how this works in the mushrooms, it would be very nice to have this yellow color. However, it's very difficult to isolate it from the mushrooms because almost everything of it is straight away further metabolized towards the red compounds, and you would not be able to get a sufficient amount of these yellow compounds, while with our SynBio approach, we can tailor the pathway exactly how we want it by expressing different enzymes. We can stop at a certain part of the pathway, or then also express different enzymes to modify the structure further. So at this point, we had a few strains where we can reliably produce these yellow intermediates, and we decided to take a small step sideways in our research. So now we were thinking, okay, we need to improve the production yield of these compounds, and we want to improve our production strain. So we aimed to optimize the yeast metabolome in a high throughput manner using the VTT biofoundry. So we have our yeast metabolic models plugged in the pathways, and then based on how this works, we can then say, okay, optimize either for biomass production or color production, and then look at the genes that are differently regulated, and those are our genes of interest. So from this list, we selected 41 genes, and these 41 native metabolic genes we expect to be involved somehow in this production pathway, providing, for example, precursor molecules or supplying cofactors for the enzymes involved in the pathway, either directly or indirectly. So then these selected enzymes were one by one rewired at 4 different expression strengths. And for this, we use our own VTT synthetic expression system to also fully uncouple the expression levels of our genes of interest from any native cellular regulation. And this provided us with 328 metabolically rewired pigment production strains. Then these strains were tested in high throughput manner in our biofoundry, where we monitored both growth and also pigment production, again with LC-MS. This data was then fed back into our models and used for further target optimization for later steps. So to show a quick snapshot of some of these results. So here on the left, we can see a plot of growth versus pigment production with our parental control strains highlighted in the pink circle. And we can clearly see a few different phenotypes where some modifications have really a detrimental effect on growth in the bottom left, and thus also on pigment production, because if the strain doesn't grow, it cannot produce anything. Then we see some phenotypes more in the top half of the panel, where we see an increase in pigment production, but also a slight to moderate decrease in growth. These in general, these modifications are still interesting, but we have to keep in mind that if we would combine too many of these modifications into one strain, that the final fitness of the production strain might not be relevant enough anymore. And then finally, we see a phenotype around the circle that the parental strains where not much is happening. So there's maybe a moderate decrease or small decrease in growth or an increase in pigment production. And these small changes are also quite expected, as the yeast metabolome is quite complex and very well and tightly regulated. So meaning that some of these changes we make will then be counteracted by the cells by just using a competing pathway to undo our changes. Then on the right side of this slide, we see an output from one of our models. And these models then give us targets like, hey, these genes or these modifications are the most relevant to increase your color production. So still a little bit about what is up next as a conclusion of my talk. So we are currently, of course, further developing our production pathways for the quaternarious anthraquinones through oxygenase discovery, because we want to reach, of course, the red compounds, which are most interesting to us. In addition, we expect to soon release more information about other tailoring enzymes like halogenases, glycosyltransferases. And then with our biofoundry-based strain improvement, we are now working on completion of the second round of our DBTL cycle, followed by a larger scale validation of the metabolic rewired strains. We have a great team working on this topic. The anthraquinone pathway discovery work from the beginning was mainly done by our PhD student, Paulo Manjetta, while the biofoundry work is led by Mari Nusanen and Emily Bennett. And that's all I have for you today. Please be in touch if you are interested in any of our work and would like to discuss it further.
Great. Now we have Karin. Thanks. Let's have the next presentation.
Good. Can you put my presentation on? Excellent. Okay. Thank you very much. Yes, my name is Karin Fleck, and I'm the CEO and the founder of the Vienna Textile Lab. And our mission is to research and develop and produce textile dyes and pigments derived from microorganisms. And this is our goal, to successfully combine them, so that they're sustainable, they're safe, and that they match the performance of synthetic dyes. And this is all what you ever hear of my pitch today, because I'm actually talking today about something that is really dear to my heart. We have a lot of projects in the past and also currently running, and they never get the visibility. I cannot talk about the industrial clients that we work with, but I will talk today about the small co-creation and collaborations that we have with other startups, with other companies, but also large research projects that are happening right now in this field. So a very small project and also very dear to my heart is the Atlas of Sustainable Colors. It's by now a platform started from Julia Caleta, who wanted to bring everybody together who produces natural colors, so people can actually access a sort of information about these companies, because they're not so easy to be found. And it was also a really nice project in the sense of we were tackling, okay, now you have this company on the map, but what is the sustainability behind it? And that actually providing sustainable numbers about a small business is actually quite an undertaking. Like, to create sustainable numbers about microbial dyes, it's not cheap, yeah. So we made also a sort of a procedure for small companies, like what they can do as a first step to understand impact calculations and impact assessments. And the nice thing about this, I have to move on with this. She also created during this time her master's thesis, which is available as a book, but also a digital atlas that you can see online. Another project very dear to my heart is a collaboration we had with Living Color with Laura and Ilfa. We have been always looking into the Vienna Textile Lab always was very focused on dyeing textiles, and we always had requests on, but why can't we do printing? Yeah, and this project was first of all, Laura and Ilfa started probably at the same time looking into microbial pigments that we did. So we're time-wise, but they were designers and we more the researchers. So it was really nice to combine our forces, work together, don't see each other as competitors, but actually do something and create something together. Here in this project, then we also presented that at the Textile in Frankfurt. It was very successful. We created a way to print with our microbial pigments, and we visited each other. This is one of the most dearest collaborations that we had. And then there is a project, maybe some people of you know, I don't know if the people in the auditorium know that, it's called Worth Partnership Project. I think at the momentary status is that it has been discontinued, but it had been a really interesting way of collaborating with designers. So we often cannot show what we do with our industrial clients, which is a pity. But what we can show is, for instance, projects like this, where young designers or people who want to create new materials are able to work with our pigments. Because, face it, we are chemists, we are bioengineers, we are not very creative. And these kind of collaborations allow us, with a little bit of financial backing, to do really great things. And I hope we will see more of this funding from the European Union. So this was one of our oldest projects. Here you can see Loreto and my team. And she visited us. She's actually originally from Chile. She lived then in Germany. I think by now she moved on to Spain. And she has a small brand called Anima. And she experienced in our laboratory how to work with microbial pigments. And also she herself was going to, I think, Vietnam and working with people dyeing with indigo. So she really wants to see and feel it. And this kind of collaboration was just like very enlightening, in the sense of what is maybe a mistake in our lab is for a designer just amazing design. And this is what I take away from this project, most of all, that we have to look at our colors in a different way. These were some technical drawings she did. And this is actually what we showcased on Milan Design Week in that year. So I think we are not that far off what her vision was. And it actually, I really liked the dress very much. Another question that came up in the next Worth Partnership project is, of course, when you think about natural colors, you always think, okay, natural colors go very well with natural fibers. And so the question, can sheep and bacteria be friends, is based around the wool industry in Europe, which is really suffering. There's high competition from Australia, from the UK, from Canada on high quality wool. And we wanted to sort of showcase. And this is not just a project about, can we get these microbial pigments cheap, and is it light fast and doing this? No, this project was more about, it was about gender neutrality. It was about how to design textiles, how to use wool in that context, the social part about these wool farmers, body positivity. And actually this picture from Ridwan Ravi, a professional producer and photographer, actually won the Sony Photography Award. So how often does it happen that a biotech company sort of is involved in a photo competition? So we're also really proud about this. And all the clothes were handmade. We also thought about the color itself, like what is gender neutral? We wanted gender neutrality in this project. We collaborated here with Pink Bonillo, and you have to know that Pink Bonillo is located in Hungary. And at the time, you can imagine how gender neutrality and body positivity resonated with the political system there. And we managed to exhibit that in some of the Goldberger Museum in Budapest in front of the people to showcase that also Hungarian design can be very open. So there was always this, like, when you produce microbial pigments and you come to these events, you often are confronted, but the fashion industry is not very sustainable. And yes, we don't cover body positivity with microbial pigments or without this, like, we don't design for that. And it's a big part of the story. And that was really nice about this project, that we could include that as a part of the topic. We did also some digitalization, because color is not color, and also communicating natural colors is very difficult because they often do not follow the typical color codes and color systems. So it is always a challenge also technically to showcase colors in a way that that they are that you can communicate that to the designer somewhere else if they're not sitting in the same room with you. So that was a big learning curve on this one. We were on different design weeks with this, won the Sony Photo Award. We worked with a digital studio in Budapest. It was quite a very interesting project because there was a lot of social component about this. Then there is another project we did on the Worth, and it was called Oceanique, completely different, with a French designer. She has a company called La Manufacture des Algues, and I'm sorry for my bad French. So, and the idea was here, we have a lot of microorganisms that come from aquatic and marine environments, and she works with seaweed, but very raw seaweed. So she doesn't want to have too much impact on the material that she's using. And so we were just asking the question, can we just make a collection just based on marine micro and macroorganisms? And this was also really interesting, a completely different material. And we also hosted Violaine, that's her name. She has actually a very colorful name, in our laboratory, where we were just dyeing together, working with the materials together, because it's not only about the colors, but it's also about how to get the color onto the material. We exhibited this also at the Milan Design Week. I like this project a lot because we can be very selective also the way we use our microorganisms. This project is, let's call it BioRedye. We collaborated with a Spanish company who's on the market with highly efficient dyeing machines. And Romano and Camino started this company a few years ago. And it is not just that you have microbial pigments, but you have to also think about the process and the machinery that you use it in. So how can you, the combination of a modern way of dyeing and a natural dye, I think, is for me logical. And in this project, we were able to do this. It was very successful. You can see how homogeneous the dyeing is. You can see here Camina, and you can see here Romano and our team, and their machinery and their upgrades that they're selling already to the textile industry. And we were funded by In Transit at the time, as also a European project. And I really enjoyed this collaboration because this is also bringing us closer to market readiness, to be on the market with two highly experienced individuals in that field. Yeah, here you can also say how many liters of water we were consumed per kilogram of fabrics. This is a very good number compared to what the standard is in the industry at the moment. Then, yeah, and now these were the collaboration projects which we enjoy and are very, very dear to my heart. These projects now, there are 3 that I want to introduce you to. They are all European research projects where we collaborate with different universities. This is an Interreg project called Green Chemistry for CE, for Central Europe, and under the leadership of Charles University in Czech Republic. And here we tackle something very unsexy. It's about this, how to improve the process to reduce CO2 emissions and how to use different types of solvents in our production. And it's very fundamental. It's very important. And if you never heard about green chemistry, there are the 12 principles of green chemistry. It's like a kind of a new religion in chemistry on how to reduce the impact when you already design your chemical processes. So no toxic components. You have to be atom economic. You have to use optimization and so on. So these are very, very basic instructions for chemists and engineers to improve the process and don't even develop something that is not sustainable or has a bad impact to the society. We are pretty much at the end of this project, so it will conclude at the end of this year. And it had been very interesting to deep dive into our scaling mechanism and our methods. This project, on the other hand, just started last year, also in collaboration with the university in Finland, University of Helsinki, but also under the leadership of the University of Technology here in Vienna, and for instance, Boras University is in there. And sorry, there's no graphic yet that I can show you how impactful this project is. It is called a doctoral network. And this doctoral network will include 12 doctoral candidates who will work on the whole scale of microbial pigments. So that means they will research the microorganisms that are involved in producing color. They will look at the biosynthetic pathways. They will look at genetic engineering. So Jörg, we will compete you. And they will also look at the extraction method, how to purify these compounds, but also at the application of the dyes, at the economical part, at the marketing part, and at the life cycle part. So it's a really like 360-degree project on microbial pigments for the textile industry and a tiny bit also on food industry. We are very proud that this project went through. It was very long in the making, and it will help specifically us, the Vienna Textile Lab, on moving further in this research. And so the next part, I always click at the wrong place here. Yeah, there's another project that I want to also introduce. It also started last year. It's a project led by the ETF, the Textile Institute, and also by Steinbeis. And it is about replacing animal products. So this is specifically about replacing kind of leather materials for instance, in automotive in this context. And of course, you also need colors there. So this has been something that we haven't really focused on in the past, was leather-like materials and other type of materials, because of course, as we develop our pigments, other companies, other startups are developing new materials. And of course, it makes only sense to combine these two types of materials. And then there is a last one. No, this was the last one. So this was basically, this is a bit of a screenshot what we do. it's always like, as a startup, you always have to understand that you cannot complete all the research yourself. You don't have the equipment, you don't have the space, yeah. You don't have the finances, and collaboration with universities is one of the fundamentals for us. We have not been, a bit to the history of the Vienna Textile Lab, we have not been a spin-off from a university. We really, in that sense, a greenfield startup, also maybe more a spin-in, so we spin back into university. But what it allowed us is that, okay, we had to compensate for that. And collaboration, doesn't matter if it was with universities, with research institutes, or with other startups, or with young designers. We became really, really interested, and it was really an important part of our journey, and it still is, to collaborate with people that are equally minded, they have the same targets, and are really interested in the project. And it lifts you up sometimes when, you know, things are not working in your own lab, but somewhere else. It sort of helps you to continue. Yeah. And so if you ask yourself, if you're looking for a collaboration partner, of course, we are open for business in that sense. We also like to host people here, so this is not a problem. We also like to collaborate on site. We have been doing quite a lot of capsule collections, also with companies. So I hope this might be of interest to you. And you can reach me under Hello Vienna Textile Lab AD. And as I said, we love to collaborate. Thank you.
Thank you, Karin. That's an absolutely amazing presentation, and really wonderful to see the value of partners and partnership projects. So really incredible stuff. Absolutely amazing to see the progress that you've made. We now have Iro from Fashion for Good. So looking forward to seeing how Fashion for Good is also supporting these types of partnerships.
Yes, thank you. Thanks for having me. A bit less of a technical talk, but I'm excited to share with you our mission and a glimpse of how we support biogenic colorants. I'm Iro. I'm an associate here at Fashion for Good. I am a chemist by background, so this is close to my heart. And I'm working in the processing team, so everything related to tier 2 technologies. To start, for those who might be new to our work, we are a global innovation platform working with brands, retailers, manufacturers, and founders to drive adoption of these new ideas and technologies. So at the core, Fashion for Good is our innovation platform. And the approach that we take is based on these 3 pillars. So we do a lot of scouting and screening of different innovative technologies, understanding what the landscape of solutions is. Then we validate them through collaborative projects and pilots. So together with our supply chain partners, we see what the impact, what the performance is within the supply chain. And then we support the scaling of the vetted solutions through collective action like financing, building infrastructure, etc. And we follow multiple topics, so including decarbonization, next-gen materials, recycling, waste, traceability, and so on. We actively screen and scout innovation on a rolling basis throughout the whole value chain. And since the start of our journey, we've identified over 3,500 innovations, and we've supported almost 200 through tailored support. So either bespoke support or through the projects that we do, and the pilots, etc. We are powered by a global network of leading organizations who are in the front row of shaping the direction of the industry. So we have our innovation partners, which are our usual partners who are involved in most of our projects. And then we have some project partners who are collaborators that contribute to specific pilots or initiatives through some of their expertise, resources, etc. We believe that the the industry will can only change if all of this innovative, amazing technology can be brought to scale. And this is quite difficult to happen on. They cannot achieve it on its own. It is a systems change journey. So where we are sitting, we strive to help the industry the solutions move from pioneering early stage practices to reaching this tipping point where they can scale and become commercially viable. And then ultimately help set the new mainstream for how the fashion industry operates. So at the end, that's really ultimately our goal, bringing that to scale and making it the actual mainstream way of the fashion industry. When you look at the life cycle of an innovation, of course, there's this like valley of death, as a lot of people have called it. The R&D and scaling phase face these major barriers related to pricing and technology challenges, and we want to give that support to provide to to give like the crucial unlocks that are necessary to overcome this part. So it can be commercial technology validation, impact assessment. So we also do screening LCAs, techno-economic assessments, orchestrating the supply and demand, and trying to secure funding so that we can de-risk that investment and the integration then for the brands, and hopefully reach mass adoption. This is more how we work from, like, how do we identify the problem, then moving to validation, then moving to scaling. But let's look into it more specific in the processing and where we're sitting. So also because today's focus is the biocolours. Here, as I mentioned in the processing, we focus on tier 2 related technologies, and a lot of our work is going towards the chemical technologies, including a lot of like dye stuff and pigment solutions. The problem is very clear here. Current dyeing relies on synthetic, mostly fossil fuel derived dye stuff. That obviously leads to high environmental impact, including water pollution and carbon emissions. And our goal is to reduce those impacts by enabling the adoption of the alternative colorants across the supply chain. So we work in the validation side through collaborative projects, as I mentioned. So that could be like foundational projects with multiple brand and supplier partners together. And then also together with our scaling team on some bespoke individual support. So that might look like commercialization pathways, doing cost calculations, implementation support within targeted suppliers, support on business models like distribution channels or agents in different locations, etc., and connections and so on. And from within the validation, 2 projects that I'm currently leading have been foundational to this effort, and I wanted to touch a bit upon those 2, their learnings, the insights, and these were the Black Pigment project and the Dye Stuff Library project. And you can also find some more information on these 2 projects on our website. Most of the outputs are, of course, for the partners who are part of this project, but we always also have an open source part where we share learnings with most of the industry. Starting with the Black Pigment, here the project addressed the fact that even though black is one of the most commonly used colors, the impact associated with the synthetic blacks like carbon black is very high, since they're derived by virgin fossil fuels, and they have also a lot of impact in terms of toxicity and the wastewater that they produce. And we wanted to validate the use of black pigments that derive from waste materials within dope dyeing, because we also understand that dope dyeing is a method where you can produce like really big amounts of black, so it could provide potentially a big impact within the industry. And the steps that we follow, of course, is like a stage gate approach. So we would make with the innovators, they would make the formulation. Then we do lab feasibility testing. Once that is successful, then move on to pilot scale trials and doing performance testing throughout, as well as impact assessment and cost analysis to also identify some commercialization pathways to support those technologies in the end. What we tested, so we tested these bioblacks via dope dyeing across man-made cellulosics and recycled polyester, so both work streams. And what we found was a critical proof of concept. So we found that waste-derived black pigment can replace petrochemical dyes in principle. So everything worked, and the trials were successful. And we've also identified pathways for the optimization, like scale up through the optimization of the formulation of the pigments and also the processes. Of course, there were bottlenecks, which obviously serve as an insight when you do projects like that. Some that I can share right now is that the pigment quality is a challenge. It needs also a lot of the milling it down for the particle size, the dispersion, the viscosity control were important, and each substrate needed substrate specific optimization, and also since the fiber and the yarn quality can be a bit different because of the different pigment load and because these were new processes. It is very important to also align that with what product you want to make at the end. So then you can adjust all the steps, the spinning, the knitting accordingly. So also already from the beginning, aligning the product with the quality of the pigment, the dispersion of the fiber that you have, was very important for the project. Hopefully, the learnings will pave the way for a safer, lower carbon black coloration system. And the project is almost near completion, so we will be sharing some findings with the industry soon. Our second foundational project is the Dyestuff Library, still ongoing as well. And here also the problem is quite clear. Well, there are a lot of biogenic dyestuff solutions that offer potential to reduce the environmental impact a lot. Also at the time of the conception of the project, there were uncertainties around their performance, around their scalability that were a barrier to their adoption from the brand. So then they needed some collective de-risking or trying this within the supply chain. And the aim here was to create a dye library that will consolidate all of those results, not only performance, but also metrics like scalability, capacity, the price, the impact, and so on. And our role is sitting at the intersection of research and industry. We looked a lot at the performance, but also beyond performance, understanding the practicalities of scalability and of the commercialization, like the price of those technologies. Not just the price, but like different commercial metrics in a way. We also wanted to focus a lot on the compliance, go a bit deeper than the industry, assess toxicity as well, as the classic industry compliance, like MRSL, for example, and do impact assessments and cost analysis again, as I mentioned, for the dye stuff. We used the validation process was again stage gated. So we do in the beginning the screening, understanding the dye types, the capacity of the innovators, their feedstock, etc. Then we define the specific validation route together with the partners, the substrates, what to test, how to test, what performance to assess, etc. Then we start with a chemical screening first, so thoroughly assessing toxicity and compliance, and then moving to the performance testing, the actual performance testing, so trying it within the lab and then in a pilot scale. And we want to consolidate everything in an online tool, a digital tool that will capture all of these results to support the decision making of our partners. A few considerations related to the process emerged while we were doing while we're doing the project. One was that characterization can be very useful to understand what is the behavior of the dye and the affinity to different substrates. So we're exploring now whether to include more analytical work before we start the performance testings, let's say. Since biocolorants can be complex mixtures, maybe it would be more useful to understand the dye classes, the purity beforehand, to already be more ready for the process optimization. While we put a big importance on toxicity, as I mentioned, although generally less harmful, biogenic does not automatically mean non-toxic, because we went even deeper. We assessed the toxicity profiles across multiple trophic levels. So also like primary producers like algae and secondary consumers like crustacean toxicity, which is, as I mentioned, more deep than the industry standards. And we believe that maybe developing some risk-based scenarios and models with the industry to better estimate the environmental exposure might be helpful in the future as well. And finally, the validation was not a one-off process. We really had to do a lot of iteration between the mills and the innovators to refine the performance, which is also partially a reason for the long timelines of processes of projects like this. The time is kind of running, but we had a lot of learnings that can be categorized in 3 main gaps. One would be the technical gap that includes like the limited color palette of those solutions, and issues like the color fastness and reproducibility. Of course, when we talk about these solutions, it's in like a large scale within the industry. It might not be a challenge in projects or like with smaller when it's together with designers or artists, but this is more like within the whole industry with bigger players that need like reproducibility. Then there's the integration gap. There's a lack of accurate characterization. Also the protocols a lot of times, how to use the dye, what to use within the recipe, and also how to explain to the brands, as Karen mentioned as well, that this might not be exactly what you have been like used to work with, or exactly the color that you might be expecting. So that communication is very important and was lacking a lot. And then finally, from the commercialization side, these solutions often come with a premium in price, but then the fragmented supply chains and the complex logistics add on top. So it's also important to work on how to optimize the logistics, find partners for distribution or for production in other places as well. There are, of course, potential solutions. They can be very individual as well. So working with we work with the different innovators together to identify how we can support them in these challenges. Yeah, and I wanted to finish with some kind of trends, but I'm a bit off time. So you can also have the slides after, but yeah, basically that we see a bit of a move to also not just the colors, but also the fixation around it and wanting like more eco-friendly fixation. A move towards digitization, like digital twins, for example, for the reactors, for the strain optimization, and being able to be like flexible with the feedstock or with the production, like sending the recipe somewhere else where they also have fermenters and being able to produce in different locations. So yeah, thank you very much. If you have any questions, you can reach out to me. And if you're an innovator or an ecosystem partner, you can get in touch through this link that I have there, or also through my my email. Thank you.
Thank you, Eero. Well, fantastic presentation and really great work that you're doing at Fashion for Good, really bringing these technologies into industry and supporting industry with the change. So really wonderful to see. So now we have a Q&A session, and we have already a question in the chat, but please submit a question if you'd like our speakers today to answer them. So we actually have one question already for Karin. So when we do dyeing using microbial biopigments, one of the biggest challenges is the light fastness of the dyed fabric. This is a barrier to market entry. Have you been conducting tests on this challenge, and have you reached any positive results?
Yeah, of course. I mean, this is the core question, apart from how much yield you get out of your bioreaction. The second thing you stumble upon is light fastness, and I think this is the number one concern of all companies in that field. And yes, of course, we have been testing, and we have also been improving light fastness as we go with each of our pigments. It's a long journey. It's not like a fast solution, and sometimes the solutions look very different for each and every pigment.
Great. And then we actually have a question for Eero as well. Have any of the projects that you've done with microbial pigments actually gone to market? And if so, could you tell us a little bit about the customer feedback?
Yes, at the moment there are collaborations that the innovators have been able to achieve through the project with some of the brand partners or through our manufacturing partners. It's mostly limited edition at the point, so most of them they are great collaborations, but we do want to be able to like reach that price point where this can be more implemented in like a mass adoption as well, because still the price point is a little bit higher for it to be mass adoption. There has been good feedback for those collaborations, but I can't share too much. But yeah, most of them perform quite well. And it's also from the manufacturer side to suggest the colors a lot of times, or like come with specific colors. It's a little bit harder to color mattes. But within this limited with these collaborations, these products, the colors were really, really beautiful, and they worked quite well. But yeah, still more work to get them in like a big scale.
And we have one final question. So this is for everybody, so we can hear some quick ideas from the team. But It's interesting to hear how microbial colours are developing and getting closer to real industrial use. I imagine that scaling production is one of the key challenges with technologies like this. So I wanted to ask, what are the biggest challenges in scaling microbial colour production commercially? Who would like to go first?
That's a very good question. I think also like Carlos already saying that, there's the first and second question. I think this is probably the third question you get is the scaling. I think a lot of it depends also on the production. Like if you really want to scale it, and then you have to work with the downstream processing, depending on, if you produce your colors inside the cells, outside the cells. It's going to be a whole different challenge. Of course, outside the cells it's always easier to scale it up and to clean it up from the rest of the systems. But the other thing is, of course, there's a lot of microbes trying to get the right growth conditions in a very large scale. Thinking about like, for some of these processes, you would need to go to hundreds of thousands of liters fermenters. It works fine if, like we did, you could put the system into a Saccharomyces yeast or other nice yeast, but sometimes you're just working in a host organism that's better suited for your color, but less suited for scale up. And I think that's a big part of the challenge also, what kind of production system you can use.
Any other thoughts from the panel?
Yeah, so one of the things you have to, in industrial biotech, when you develop materials, this is not just about biotech. There's the chemistry that comes after, the textile chemistry, the regulatory, and most of all, the product development. Because just because you have a colorful powder in your hand doesn't mean you have a product for the industry. And that is something they don't teach you in startup school, really, like that. In that way, how important product development is. Like, is it what kind of powder? Like Iro said, you have to grind it down, maybe these things, or add additives. And I think the combination of all these disciplines, that's each of them has a success factor. Like, how rich your yield is in the bioreaction, but then also how sustainable your extraction mechanism is, how good the textile application of the dyes are, but then also does it go through regulatory? Is there anything like a sensitivity popping up for this dye? And most of all, what type of product is it? Is it an ink? Is it just a pigment? That is, I think, the biggest challenge of all of them. And maybe all of them together are really, like, if I think back when I started, if I would have known, I don't know if I would have gone for this.
We've basically run out of time, but maybe Iro, you could quickly say a couple of words.
Yeah, no, I have to agree with both. I mean, there are technical challenges when it comes to scaling up the actual bioreactors. So the yield does come down, and that's why now we see like also these trends with the digitization, like digital twin of the reactor and so on. We'll see how that will help. But yeah, I would agree with Karen quite a lot. Also, for example, if you can bring it down to a powder, that's always more helpful to distribute it rather than having a liquid. But then it takes a lot of energy, and the emissions, the LCA might not look as good. So there's always this trade-off on, like the distribution, the production is quite important. Yeah. I mean, I could talk for some time, but yeah, I think that's, yeah.
Thanks so much. And we also had loads more questions in the chat, so we'll see if we can get back to you individually with the answer to those questions. But thanks everybody for attending today and for participating in the questions and answer section. And a huge thanks to our great panel of speakers today. So when the webinar ends in a moment, there'll be a feedback questionnaire that will pop up after you close it. If possible, please give us some feedback, and there's another opportunity to ask some questions and request a meeting with our experts. And please contact us if you have any questions. We also have a contact point on our VTT partnership team but also any of the speakers, including myself today, if you'd like to get in touch and really kind of continue the conversation. I mean, I think you've probably seen from today's session that partnerships is really at the heart of driving biocolours renewal into industry, and we're really proud here at VTT to be a key part of this and want to work with as many innovative partners as possible to make this actual impact happen in real life industries. So thank you again for attending today, and we hope to see you very soon. Goodbye.
Bye.
Thank you.